sometimes. So to be normal. So other than exosome and EVs, so we have like isoforms that carry 1 micrometer larger particles. And the other one is microton. I believe that today I'll talk to talk about the mechanism how to characterize the mechanism structure and functions. And first, I think, after considering the purity, we have to make sure what we are going to use. I think the MICEF 2023 gives us a very good example here. So we have a lot of particles, but maybe only a very small portion of these particles could be classified as extracellular vesicles, and the other ones should be non-vesicle extracellular particles. How serious is that? For example, in plasma, we can expect that 1 EV was mixed, around 10 to the 9th or 10 to the 8th particles generated by lipoproteins and other plasma particles. So that means that we have a very, very limited number of EVs in plasma. And also, the non-EV particles can carry a lot of RNA types, such as microRNAs. For example, in this paper, we can see they use, besides exclusion and chromatography, to separate a lot of fractions here. And these are the non-EV fractions. You can see that a lot of microRNAs are in power. So I think today our first talk will cover, we'll use a very interesting, technology to distinguish the non-EV associated microRNAs. Okay. But when we go to, when we consider that as purified as possible that we can get from the combination of a lot of technologies, we can, this paper showed us that if we want to use the ultracentrifugation combined with SEC, and then we have a very limited yield, maybe lost everything from the plasma. So when we pursue extreme purity, we lose almost everything. So that's a balance. So we have impurity and have extreme purity. So our group published the new paper. We propose that the purity is not always that important, especially in laboratory medicine. That means we have two bottom up and top down strategies here. So bottom up, we have very good isolation of the EV and particles. We can recover all the particle carrying biomarkers from our methods. So we can, and then if we can solve the clinical questions, that will be adequate eventually. And for the other direction, if we want to know which biomarker is specifically located in the EVs, so we need to extremely purify EVs. So both directions are very important for EV separations. For example, so in the MICEF 2023, there are many isolation methods have been proposed. Especially, we can look at these two, the precipitation and filter concentration. So these two methods will not get rid of lipoproteins at all. But it has a very good yield. The recoveries are very low. But if you look at the ultracentrifugation and the density gradient ultracentrifugation, and we can have a very good purity, but still will lose a lot of particles there. And the other methods, such as SEC and affinity-based methods, these the SEC will never avoid co-isolating contaminants of the level proteins because of the similar size of these particles. And for IP, we can only have very specific subtypes of EVs. And other affinity precipitations will have different problems because of the protein and other particle have a very similar features to EVs. And this one I will not introduce here. So then, when we consider the purity, I believe it's a moving target. I cited all the quantization methods from my side. So we can rate the quantification of particle number concentration. It's a very instrument dependent. So a lot of instruments will have a 10 to the 6 magnitude difference. So we have to take care of pay attention to that. And particle size will give us only size. It's not the memory, biological memory structure. It will never tell us for that. And the quantification of total protein is fine. And total lipids, we know level proteins are lipids. And total RNA is fine. But EV morphology will give us a snapshot of the electron and the microscope. And if we want to use the protein composition to define EVs, it's almost impossible. There are no specific EV protein markers for all type EVs. There's no specific protein markers for all type EVs, according to the multiple consensus. And also, particles are not equal to EVs. So because we use a lot of particle protein ratio to define the purity. But that one will, if the particle itself is not EV, so if you use that ratio to quantify purity, it's a problematic. Another method is very interesting. This method was established by Professor Ye Jianwei from Xiamen University, China. They use the Triton X100 to disrupt the EV structure because the EV has a biological membrane that's molecular lysate. So these particles will disappear following the Triton 100. So this is an example. For example, this one is EVs from cell supernatant, isolated from cell supernatant, cultural supernatant. So 96.0% of the particles can be laxed by China 100. So it's I think it's a very definite answer to the purity of these, for these EV. But if we have a very large amount of lipoprotein in the sample, this method will never work because China 100 is walking out. It will disrupt the lipoproteins into small particles. So we will never get correct result. That also echoes to my previous slide. So particles are not equal to EVs. And the other thing we need to consider is are co-isolating contaminants really contaminants? For example, in this paper, we found that in the SEC method or UC method, a lot of albumin and lipoproteins are constitute of major contaminations. But the top ones contaminants are antibodies, like IgG, IgM, and a lot of antibodies. This confused us for a long time. I think that is did a very good job. So we use the THP1 cell culture to produce naked EVs and then put them back to the plasma and see what kind of these EVs capture from the aggregate. So we can read that a lot of immunoglobulin was captured by these naked EVs. THP1 cells are monocytic, so it has a lot of Fc receptor. So it's very reasonable that antibodies can not specifically bind to these EVs. So, and these naked proteins can capture other known contaminants like albumin and also a lot of lipoproteins. A lot of lipoproteins too. So it's I think what they call the EV corona should be very important information for our future study. So very consider so then we should really think about the contaminations. So that's the bi-direction strategy for that. For example, for this is the case. This is the case of the adequate enrichment. So 10 years ago, we invented products that can capture the proteins called as and we know per Dr. Zheng Wei and Zhao Kewei's group, they found that CA125 is exclusively in the extracellular particle fraction, so not in the closed loop. So it's when they use this device to test the ovarian cancer, they got the diagnostic powers from 0.091 to around 0.98. Okay, so it's a very important application there. And also, this is SODEX is using only the centric ultrafiltration can do the diagnosis of prostate cancer from urine. And also the purified EVs can do a lot of cancer subtypes, like this paper show. A lot of cancer types, 7 of them can be distinguished by EV proteome. And also it's good for the top-down strategy for EV-based EV subtyping. Is also good for breast cancer stratifying. And also we have a very good scoring system. Okay, so we have a very interesting conference about the Chinese herbal medicine will be held in this fall, August. So we invite you all to visit Guangzhou, China, to give us a talk and join us to discuss some plant EV research. So I will use 3 minutes to give the introduction about the upcoming amazing talks. So Dr. Xinyang Liang from Nanyang Technology University will give us a talk about a very new method that can both quantify microRNAs and surface proteins. Especially, as I said, they will take care of the non-EV associated microRNAs too. So it's very interesting talk. And Dr. Xu Chong from Southern China University from China will give us a very interesting talk about the bacterial for colorectal cancer diagnosis. So they optimize a very good method and standardize the isolation of these EVs. So it's good to know. And Dr. Ouyang Su from the University of Toronto, they use a very good, I think it's aqueous by phase system to establish a very, very fast EV research. Okay, there's rumors that BTS will disrupt a lot of EVs, but I think for the mRNA-based lipid biopsy, it's very good also. It's very fast and cheap. And Dr. Tony Heise from the Ohio State University will give us a talk about how to distinguish EVs that on the extracellular matrix. The extracellular matrix and EV interaction by using this light-induced extracellular particle assorption. And finally, Jia Yu from Nanyang Technology University, Singapore, will give us a very interesting tangential flow filtration on the chip. So it's and they have very special structure that are higher known microstructures. They can have a very fast extracellular vesicle isolation and from the blood plasma. So let's welcome all the audience. So I will turn the host to Dr. Hamdi. Okay, thank you.
Thank you very much, Professor Tan. The talk, it was really nice presentation. And now I would like to welcome our first speaker, Shen Yuan Guo from Nanyang Technology University.
Isolation of complete platelet-free plasma for enhanced detection of blood EVs, microRNAs, and also the surface protein. So blood EVs are very promising biomarkers to various diseases ranging from cancers, neurodegenerative diseases, and even metabolic diseases. Translation into clinical setting is still very challenging because there are many steps that usually we don't find the problems in the laboratory. During scale up in the clinics, then all the problems will surface, which require a lot of attentions and effort to address. So I would like to start with the typical clinical workflow on how blood EVs will analyze and generate the diagnostic report. So usually, blood will be drawn in either clinics and hospitals. So depending on countries and hospitals, the blood may take up to 4 to 24 hours to reach the labs. So here, so very often the vibration or the changes in the condition during the transportation will cause hemolysis, platelet activations during these steps. And commonly plasma are extracted using centrifugations, which remains laborious and they are lack of standardization across different hospitals. And sometimes we do found residue cells and platelet in the plasma. So this will actually create high variability, especially in terms of the pre-analytical aspects to the circulating EV and RNA research. So we think that there is an unmet need for an automated and rapid blood plasma isolation so that we can exclude the confounding factors from the cells and the platelet. So recently there are a lot of publications trying to address the residue platelets in the plasma. So these platelets usually very hard to be detected using hematological analysis, but it can be detected using more sensitive flow cytometers. So some methods have been proposed using a filtration after plasma isolation to remove and using modern device to streamline the entire process and prepare a better plasma for EV analysis. So for these problems, we have developed a microfluidic chip, we call it EXOA, for platelet-free plasma isolations. So the device is very simple. It has a 2 inlet. So the sheath and the blood can be pumped into the chip using power. formed inside the curved microfluidic channel. Larger cells and platelets can be pushed towards one side, whereas the smaller particles like EVs and plasma can be collected on the other side. So in the high-speed images, we can see that all the cells are being repelled to the downside, which we call it outlet 2, so we call it the waste outlet. And plasma and EV can be collected at the top outlet, which we call it outlet 1. So we designed a cutoff size to 500 nanometer, which means we can exclude particles larger than 500 nanometers. So it's pretty simple, it's a single step, and it does not require a centrifuge. So we hope that it can help to increase the reproducibility of the plasma. And also we can process 1 ml of whole blood in 10 minutes. And because the pump will only draw a small amount of blood at every moment, so the blood basically stays less than 1 second throughout inside the chips. So it's compared to centrifugation, which the entire blood tube will sit in the centrifuge for around half an hour to 1 hour. This greatly minimizes the shear exposure time to the cells. So not just blood, there are a lot of platelet-rich plasma already stored in the viral bank. So we also look into it. So we look into the platelet pool of plasma, which is planned by the viral bank. So when we pass it through our device, we still can see under the microscope all the residual platelets. They will form a band, and we can exclude them to the waste, so leaving the plasma unscathed. So we further characterize the residual platelets in the plasma and use flow cytometry and compare it with different centrifugation protocols. So the exo accumulation is the orange color. So you can see that different centrifugation protocols we have quite a huge variance in the concentration in the plasma. So we choose one to compare. We found out that the platelet-associated microRNA can vary as much as log 2 FC up to 5 times. So this is quite a considerable difference in terms of microRNA analysis. We also checked the RNA size, and we realized that those cellular RNA size, which is larger than 200 nucleotide, are not present, which means it's kind of show of a very good cell removal during the isolation process. So to characterize the EV isolated using XOR plus SCC, because we want to remove the background proteins, so we compare the EV in different isolation protocols. We also benchmark with the conventional centrifugations and effect expression chromatography. So for better visualization, we color code to different colors. So first we analyze the EV markers. We use CD9 and fluorescent nanoparticle tracking analysis to compare the size distribution. We found that XOR and XOR plus SCC, the EV are all below 200 nanometer, which remain the size range. So if we do a statistics, we can compare. We found that XOR will help to reduce the EV aggregations. And if you compare the concentration, most of the EV exists in the plasma. And during the isolation, there are some losses. But the losses still make the XOR plus SCC as 6 times higher yield compared to the UC. And also we quantify the proteins in the isolated EVs. We can remove more than around 1,000 times reduction of the background proteins to around 100 microgram per ml in the remaining EVs. As you know, the albumin is one of the lipoproteins, consists most of the protein in the plasma. So a proper protein removal is required to make sure the EV has better purity and better consistency during the downstream analysis. So we also do some imaging to check whether the morphology is still intact. So we can see that compared by the arrow, we still can see the culture of the EV. So we also study what happened to the culture mediated EV after the isolation. You can see that the EV still remains small and there's no sign of aggregations. And under cryo-EM, you can still see the lipid bilayer of the EVs. So we also try to analyze the surface protein on the EVs. We first use a fluorescent nanoparticle tracking analysis. So we use a platelet marker, monocyte marker, and neutrophil marker. So we are lucky to see that we can detect SARS among healthy patients and also type 2 diabetes patients. And we also tried nanoflow cytometry. So this is a screening data, it's still in progress. You can see sign on CD9 markers. and we take some of the key selected which is data activation markers. So we'd like to see whether we can continue and construct the pipeline of plasma in and plasma plus flow cytometry to give a quick analysis results. Well, flow cytometry is not easy, especially if you use plasma as an input sample. So I think we will need more work to make it more robust. So other than the surface EV markers, we also look into the microRNA. So we are quite interested to know, is it a total microRNA in the plasma, or EV specific microRNA can bring us a better diagnostic performance? This is why we do a comparison. So most of the microRNA are shared. Well, EV, what we call N1 plus SCT isolated EV, has some distinct microRNA that is not found in the total plasma or is not enriched. So if we do a differential express microRNA analysis, we found that they do have individual panels of differential express microRNAs. So we have quite a small cohort around. So we would like to repeat these studies and confirm which microRNA actually is more robust and differentially expressed in the cohort. So here we're using a microarray qPCR based. So we hope that it can accelerate the translation next time. So to repeat the number using a RT-PCR. So we also try to see whether we can help to reduce the platelet contamination in the serum, which is already processed and banked. So we took out the serum, we thaw, and we run through the device and compare to centrifugation. In the highlighted yellow panel, you can see that there's a relative abundance of the CD41, which is a platelet specific, that can be reduced by using this. So our hypothesis is it could be the process, our chip provide a more gentle separation, which help to reduce platelet fragmentation, or it provides a better platelet depletion during the isolation process, which helps to also reduce the platelet-derived EVs. So all these devices require high throughput and automated to be implemented in the clinical settings. So that's why we build a prototype to demonstrate the automations. While this is not good enough, we're also working on the next generation to faster and easier, and also high throughput and multiplex. So the concept of this machine is to make blood processing single step, easy to use, and can provide a cell-free and platelet-free plasma in front of test setting. So meaning say after blood draw, blood can be immediately processed and converted into a plasma, which is much more stable, easier for transportation, and hope to increase the consistency of the EV diagnostic assay. So the chip is very simple. It can be converted into medical plastics without membrane, electrodes, so we hope it can drive the post-flow or messy adoption. So we also look at the cell culture, so we build a DIY digital point system to inside the biosafety cabinet. So we can extract the EVs or cross cell-free layers from the culture media, and we do a tracking of the EV secretions. So basically just take 1 nanometer from the culture flask, process through the chip, and measure the signal expression. So track the dynamics of the EV secretion and also measure the size difference. We also have our own chip, so we hope we can fractionate small EVs from large EV, or also can fractionate small EV from bacteria. We hope that we can explore both types of particles for biomarker analysis next time. So in the future, what we will be build a computer automated streamlined methods so that blood in, EV out, and advanced we have on-chip EV detections. So the micro TFF, initial contribution later in the later session. So please stay tuned and listen to this interesting talk. I'd like to acknowledge the funding sponsors and also my supervisors. So we are welcome for any collaborations and feel free to discuss how can we improve our work. We are happy to take a share. Thank you.
Thank you very much, Dr. Shek. We have no questions
Xin Zhang from Duke University, USA. So very good talk. I just have one question or comment. So as I understood from your study, you use CD41A as a specific marker for platelets. But as I understood, so this marker is actually also expressed by hematopoietic stem cells. So then I personally do not think this is an exclusive markers to identify platelets or platelet-associated genes. Do you have any understanding or any explanation for this part?
Okay, yes, I agree. CD41 is not very exclusive biomarker for platelets. It also expresses other cell types. I think in future we need to include more platelet markers like what I want to say, the CD61 here, so to present a more exclusive categorization. Yeah, you're right. So I think you'll be able to add this into our future essay. Yeah, thank you very much.
That was a great talk, thank you. Looking into the future, I do see that you are trying to integrate even EV isolation with creating platelet-free plasma. So when it comes to EV isolation, I mean, lipoprotein particles and many other particles, which are trillions in number in your of blood, do you think the microfluidics could somehow be designed, even if they do have an overlapping size, that you could gate one versus the other?
Okay, yes, this is one of my dream lists. Yeah, for microfluidic device, there's a huge trade-off between size resolution and speed. Right. So if you check the literature, you can find it's a very fantastic. They can separate lipoprotein from EVs, but they're usually quite slow. For example, one tube of blood, they will take hours, and clinics cannot wait so long. Yeah, it's very interesting. We also receive these comments a lot. Yeah, we are working very, very hard to make it happen. Yeah.
I'll talk to you afterwards. Thank you, great talk.
Thank you.
Thanks, Dr. Shen. I hope this dream comes true. We all do. Our next speaker is Dr. Yikong Zhu, and from Southern Medical University, and his talk is on optimized isolation of fecal bacterial extracellular vesicles and its application in colorectal cancer diagnosis. Her talk is on optimized isolation of fecal bacterial extracellular vesicles and its application in colorectal cancer diagnostics.
Hello, dear professors and UV research partners. Good afternoon. I'm Shi Yitong, a master's student from Nanfang Hospital, Southern Medical University in China. Thanks for the valuable opportunity provided by us with this valuable opportunity. And it's a great honor to be here to share. And my report's content is construction and application in colorectal cancer of isolation method for bacterial extracellular vesicles from human feces. Now I will mainly report on the following 4 aspects. According to the Global Cancer Statistics Report 2022, colorectal cancer is the third main disease among all cancer types and is the second leading cause of cancer-related deaths, posing a serious threat to people's health. And in recent years, intestinal flora has been recognized to be closely related to the development of colorectal cancer. However, the key components of gut bacteria that act on the body remain unclear. With the increasing understanding, the important role of bacterial extracellular vesicles, a kind of nanoparticles mainly secreted by bacteria, with diameters between 20 to 400 nanometer and carrying bioactive molecules such as proteins, lipids, or nucleic acids, etc., imitating the biological functions of bacteria is more and more emphasized. However, given the diversity and complexity of their bacterial origin, secretion, and carrying functional molecules, the heterogeneity of EDVs in the gut is not clear. Only by elucidating this issue can we help better to understand the mode of action of intestinal bacteria and thus provide new solutions for diagnosis and treatment. So in order to gain a deeper understanding of EDVs in terms of their types, quantities, and carrying functional molecules, so the first key challenge to be addressed is how to isolate EDVs from the complex intestinal matrix. Feces is a commonly used biological sample for characterizing gut bacteria. However, given the limitations of single separation methods and the complexity of fecal samples, there is currently no uniform standard for fecal EDV separation. Therefore, there is an urgent need for an efficient and convergent standardized strategy for EDV separation. As a result, our study consists of the following two parts: constructing a method for separating fecal EDVs and exploring the correlation between fecal EDV and CRC. First, we optimized two one-step separation strategies, which were density gradient centrifugation and size exclusion chromatography, EDC and SEC. We optimized the buffer conditions, centrifugation modes, including top-down and bottom-up modes, and the centrifugation times for the EDC strategy, using the 6 kinds of bacterial EDVs as positive controls, and uniformly choose the aldextrin Higgs buffer with 7-hour centrifugation as the final condition. Results such as fluorescence intensity and protein characterization analysis show that EDV were mainly informed in fraction 6 to 8 with a wider distribution of negative EDVs, and positive EDVs were enriched in the higher density fractions, suggesting that it might be related to the bacterial characterization. Then we evaluated SAC strategy with 20 milliliter column, including CL6B bacterial as positive controls. Bacteria 7 were uniformly selected as the collection fractions for subsequent studies. After optimizing the DTC and the SAC strategies and conforming the distribution of bacterial EDVs, we used both strategies for fecal EDV separation, and we found that fecal EDVs had a similar distribution range as bacterial EDVs. And that the purity was higher after isolation relative to before isolation. The results of the interference assay show that EDC isolation strategy can exclude some contamination with interfering proteins. And eukaryotic extracellular vesicles, which may be enriched in fraction 5, characterizing by CP63 and synchrony, conforming the methodological reliability. In the DTC system, the purity of EDVs in fraction 6 to 8 was as high as 80%. However, in the SEC system, we find a significant increase in particle recovery when the grams of feces extracted increased, and we find that it might be related to an increase in the lipid content, and therefore recommend that the mass of feces extracted by SEC should not exceed 4 grams. Next, we evaluate the separation efficacy of one-step and the combined separation methods, really including 5 methods: SEC, top-down or bottom-up, TGC combined with SEC, or combined with neutral centrifugation. In terms of morphology, particle size, and protein markers, protein and recovery, and finally we find the optimal strategy for fecal EUA separation. We find that the morphology and particle size of EUAs extracted by different methods meets definition, and the stack method had higher recoveries. However, the TGC combined with stack method resulted in higher purity. The combination isolation method had higher levels of or LTA and lower levels of mutant proteins. Given the higher particle recovery and higher levels of markers, we finally choose top-down TGC and SEC and the subsequent fecal EUA separation method. And in part 2, we explore the correlation between fecal EUAs and the CRC. Based on a multi-center platform, we collected fecal specimens from healthy individuals and CRC patients and separated fecal EUAs. First, we explored the effect of free nucleic acids outside the EUAs on the sequencing results and found that they had no significant effect on GRAS or DNA sequencing analysis. Next, we found significant differences in combination of gut bacteria and EUAs. At the phylum levels, EUAs were predominantly derived from bacteriodata, and the EUA secretion was reduced from formicules and actinobacteria, suggesting that EUA secretion may be related to bacterial characteristics. In CRC patients, bacterial diversity was significantly reduced. Although there were no significant changes in DNA diversity, the combination of intestinal EUAs was significantly different from that of healthy individuals. And the combination of intestinal bacteria or EUAs showed dynamic changes at different disease severities. At the bacterial level, compared to the healthy population, CRC patients had significantly higher proteobacteria with a significant increase in bacterial Staphylococcus and Common and a significant decrease in health-related dendrils such as bifidobacterial and Eubacterium. At the level of EUAs, there was a relative increase in EUA secretion from signals such as Common and Lotia, and a relative decrease from dendrils such as Eucalibacterium and other important intestinal cornerstone bacteria. Further, by constructing a random forest model with RC analysis, we find that incorporating fewer genus-level intestinal EUAs could achieve similar CRC diagnostic efficacy as that of bacteria, suggesting that intestinal EUAs was equally important, and that the variability of the intestinal EUA composition and abundance is expected to be a potential indicator for CRC diagnosis. In the future, we will further focus on the species or stream level and excavate the potential pathogenic bacteria or probiotics, and explore the specific mechanism that specific streams influence the development of CRC by secreting EVs, so as to contribute new solutions for the early and precise diagnostic or prognostic risk assessment of CRC. Finally, thanks again to HDB for this valuable opportunity, and I would like to thank my supervisor and all the teachers in the laboratory and the help of all the lab partners. Thanks for listening and welcome any feedback and suggestions. Thank you.
A really exciting presentation. We have time for 2 questions.
Hi, Hong Leo, University of Toronto. Very interesting question. Presentation. I have an interesting question for you. It's kind of childish. Is there a smell difference between the PFAS versus bacteria? Never did a smell test, I guess. Oh, it's fine. This is a childish question. But my real question is, did you control for BMI? Like overweight individuals within the cohort? Like, because we know that, how do I say this? Higher BMI individuals have a different gut flora. So did you control for that?
And you see that flora of BVs, our isolation method,
Well, I mean,
dispersion efficacy.
well, we're probably discuss afterwards.
Okay, thank you.
Anyone else? I have a question. I just wanted to know, because we keep talking about the gut microbiome, particularly if it's upper gut or lower gut, it changes. So do you have any thoughts about going further and looking into different gut flora and then trying to isolate TVs depending on what?
You see, if I didn't mistaken, I want to asking, you want to asking the how I can isolate the fecal debris from the feces.
Yeah, like we can talk about that later after this because we don't have time. So okay, I'll do that later on. Okay, thank you very much. No problem. Our next speaker is Boyan Su from University of Toronto Sunnybrook Research Institute, and he will be talking about a low-cost kit for gentle, effective, and timely extracellular vesicle isolation, accelerating development of RNA-based liquid biopsies for neuroimaging.
Well, thanks for the introduction. So let's get started. So Dr. Wang has already shown this slide. So we're just showing this section of work for MySat 2023 period of EV separation and concentration. But I'm sure everyone who I think the most important take-home message is that there's a bunch of diverse methodologies to achieve EV isolation. And there's no universal one or the perfect one, so we have to choose the appropriate method for the isolation. It depends on what kind of research question we want to ask. So the method I'm going to present today is the method of developing and optimizing using special fluidics called the equals to the ETPS. So to do that, we simply mix the EV containing biofluids with these two polymers, the polyethylene glycol and dextrin. So these two polymers are water soluble, but under a certain composition, they're invisible with each other. So when we mix them with the EV media, we simply swing them down by 200 times G for 15 minutes. And this separation status could reach. So I just want to point out, 200 times G is a much slower application speed. Very rapid EV isolation. bottom dextran phase. And we can simply separate the PEG phase and dextran phase. And we expect the total EVs going to the DEX. Okay, so we want to analyze how the EV is being partitioned in the system. We're using the flow cytometry analysis machine from topology. So when we're flowing the conditioned media from PC3 de-escalating cell lines, this is a prostate cancer cell line that we transduced with lentiviruses for Z-escalating extraction. This is just a brighter version of green fluorescent proteins. So in this case, we successfully detected the green fluorescent EV subpopulations. And at the x-axis here, we see the large angle light scattering, which could represent the relative size of the EVs we can detect here. So the left edge relative has a size of 100 nanometers, and on the very right edge, we have 1.3 micron-ish. So once we process this cell culture conditioned media to ATPS, we didn't see any emission positive EVs at all in the PEG phase. Well, the most of this de-escalating prostate population has been enriched in the bottom dextran phase. Another advantage of ATPS is that once we got the dextran phase from the first phase separation, we can simply mix that with additional PEG to achieve a second round of phase separation. And to do that, we can improve the EV enrichment. As you can see, the second dextran phase has lower volume. And also, to do this, we can improve the purity of the EV samples. So the quantification, we've seen the EV concentration within the second dextran phase is greater than the first dextran phase, or also greater than co-precipitation method. We also access the EV efficiency. We see that in the second dex phase, the EV recovery remains very high. So until this slide, the story for ATPS for EV isolation sounds very beautiful and amazing. But actually, this method is not a well-known method yet. And we believe the reason why ATPS hasn't caught up is due to two major challenges. So first of all, once we got the EVs isolated in the dextran phase, as long as we add alcohol in the system, we start seeing this precipitation form immediately. This is due to the alcohol dextran precipitation process. And sadly, for all of the RNA isolation methods that we can find, pretty much like they have alcohol precipitation step. So alcohol precipitation here is precipitating RNA. But once the RNA is co-precipitated with this dextran, it's going to give us minimal yield of RNA, and also having this highly viscous dextran alcohol precipitation is going to interfere with downstream analysis, such as qPCR or the sequencing. The second challenge is that we can't even do flow cytometry on those EVs isolated by ATPS, and just ATPS after immunolabeling. In this experiment, we simply mix the cell culture conditioned media with different concentration of dextran, and we label them with antibodies targeting the EV biomarker everyone's using, CD9, CD63, CD81, and flow them. And we find that the concentration of the phosphate shows this dextran dependency and can plateau at 80%. And this is consistent along large EVs and also small EVs. And in this cytogram, we're showing that as long as you add dextran to your conditioned media, label them with antibodies, you start seeing this boosted population for the target EVs, but likely due to the non-specific binding between antibodies and EVs. So we have to find a way to get rid of dextran. Our solution is using this enzyme called dextranase. Break down EVs to simpler saccharides such as isomaltose or other oligonucleotides. So dextranase could be isolated from multiple fungal species such as penicillium or Catonium. And it's been used widely in sugar industry and dentistry since the 1950s, and the major enzymatic activity being addressed on this hydrolysis reaction on this 1-to-6 linkage within the dextran branches. So we first access validated dextranase treatment is going to cause damages on the EVs. So as you can see here, we have the samples isolated by ATPS, and we treat them with different concentration of dextranase. we've seen no reduction of the concentration of this ES1 positive disease population, and also the distribution of the population remained the same. We also performed the EM to see the residual structures, and we find that the structures remain intact. So definitely doesn't damage these at all. And then we process these samples to triacylchloroform-based RNA isolation, and we finally see it's working. We can get some RNA out by seeing this clear solution here, indicating the complete depletion of the presentation. And we also find the sweet spot for the complete depletion, which is this 0.5% dextrose treatment. Okay, then we measure the volume of this dextran depletion, and as you see here, as long as we use dextranase at 0.5% and even more, we see this complete depletion of dextran precipitation. And we use bioanalyzer analysis to analyze the RNA content from being isolated. And the interesting finding is that no matter how much dextran precipitation you have, as long as you have a little bit, you don't see much RNA yield at all. And once we completely depleted that, we're seeing this RNA concentration, we're seeing this massive increase of RNA that we can isolate it from EVs, being isolated by ATPs. And now we get some RNA out. We want to see if we can do some downstream analysis, such as qPCR sequencing. And we also want to compare our method with some other methods, such as ultracentrifugation, the gold standard, and also the QIAGEN AXL RNA easy kit. This is a very widely used kit for RNA isolation on EVs. So we're trying to isolate the RNA from the conditioned media from these two cells, the PC3, the prostate cancer cell, and the breast cancer cell, NDNB231. And we find this very consistently. Our next generation ATPS isolated more RNA than PI2. And we perform small RNA sequencing, and we have successfully detected microRNA, circular RNA, and also small nucleolar RNA. So for microRNA, we've seen this good overlap between ATPS and ultracentrifugation. It's slightly better than QIAGEN. And we also, for the circular RNA, the overlap remains very high, about 87%. And for the slow RNA, the good overlapping remains. So in summary, our next generation ATPS is equivalent to QIAGEN in terms of small RNA analysis, and also our methods very representative small RNA column. I just want to recall, we previously have shown that as long as you have dextran in your system for flow cytometry analysis, you have this non-specific immune labeling. Also once you isolate EVs using ATPS and you label them with antibodies, when we're calculating the recovery efficacy, we're seeing this abnormal 200% recovery. So where are those additional signals coming from? Right? It's pretty much due to the non-specific antibody labeling. Not surprisingly, as long as we treat them with dextranase, we reduce this recovery efficiency to a reasonable level. And our next generation ATPS system also shows very high recovery efficiency, close to 100%. So all of the stories I just shared in the previous slides are coming from on cell culture condition media. And we also want to test whether this method could be applied on human samples, such as plasma. So compare our method with 3 commercially available kits from Norgan, from Richard, and from QIAGEN. So in the flow cytometry analysis, we've seen that our method, along with the enriched and total exosome isolation reagents, they are showing a very representative positive population. In this case, CD9 positive EV population. This population is being enriched, and also the distribution is very similar. For QIAGEN, it's kind of favored to the larger EVs, and for the Norgan, because they have a filtration step, they preferentially isolate small EVs. And this find is consistent across CD63 and CD81. In our recovery efficiency analysis, we find that ATPS and thermal all very good in terms of isolating total EVs. The next generation ATPS method is better than high energy, but also low. So in summary, we find that ATPS is a very gentle, effective, and timely method for EV isolation. But we can only get RNL with the help of dextranase, and also the dextranase could also prevent the dextran-mediated non-specific EV antibody label. So at the very end, I want to thank my supervisor, Dr. Hong Leon, sitting up there, and also all of my colleagues from the lab. I also want to thank Dr. Scott Tsai and Dr. Matteo Gianni for bringing ATPS method to us, and also Luis helping me on this project. I also especially thank ISAP 2024 for giving me this opportunity to share this project, and also reward our work using the Crystal Scholarship. Thank you very much.
Thank you, Gwen. We have time for one question.
Thank you very much for that presentation. It was very, very nice. I just have a small technical question. So when you stained your EVs, did you do single staining or did you do triple staining?
We have to both. Like we only do so for the data I'm showing, like all of the antibodies are conjugated with the same fluorophore, but we have also performed let's say double labeling CD9, CD3 positive with different fluorophores.
Yeah, because we have the allergy, and every time I try to do double staining, it seems not to work for us at least. So I was wondering if you're able to do that.
Yeah, for the plasma samples, we do see like very limited double label events actually, but from the cell culture condition, yes, no, they work better. You have to take the appropriate antibody and also appropriate fluorophores.
Yeah, and you were also doing from biofluids, so that's, yeah. Okay, this is it.
Thank you very much, Graham. Our next speaker is Dr. Colin Hassie from Ohio State University, and his presentation is on light-induced extracellular vesicle enlargement.
All right, hello everyone. I'm Colin Hassie. I'm at Ohio State. I'm an NIH mosaic scholar in biomedical and chemical engineering. I work with Dr. Eastwood primarily, and I work with Zach Schultz as well in chemistry. And very lucky to be starting my lab in September at Western University in biomedical engineering. I'm going to talk about a new technique we've developed called light-induced EV absorption. I just want to thank the organizers for the invitation. Thank you for being here at 5 o'clock on Saturday. This is going to be very qualitative, lots of beautiful images and videos, so you don't have to just kick back and relax. So just briefly, why did you go about this technique? I cared because I became really interested in microsomes, which are one of these subtypes of EVs that can produce an embedded on the ECM, deposited by cells, and then can be internalized by other cells. But I think regardless of what type of EV study, if it interacts with the ECM potentially, it can be a very dynamic process, and if it interacts with different materials in certain ways, and it's relevant to your research, you can just think about how this would apply to your specific EV subtype. Thanks to the introductory speaker and our session chair, I can kind of skip this slide. Basically, my lab uses all of these techniques interchangeably or independently, depending on the starting material we use and the downstream application. We can use them to the context of the specific experience. But in this presentation, I'll be using them kind of interchangeably, just assuming you know what we're talking about. So I thought it would be kind of cool to go back in history a little bit. So these are two papers I found in 1944 and 62. So hopefully you enjoy this. This is they call them micro extensions, and at the time they still called them retraction fibers. But I think these are the first examples of someone the imaging microsome. So if you look where I put these arrows, you can see with these different cell types. Like the one on the left is one of the first TEM studies of cells ever, and you can see these kind of EVs at the intersections of these retraction fibers with the tips of them. You can see them here too. So I think it's kind of interesting that people have been imaging these for a long time, and just I think didn't have a way to study them. But if you go back and read the papers, they call them out directly. I became interested more recently, largely inspired by Li's work at Tsinghua. Video start. So the top 2 videos there are from this group, particularly this, my cursor? Yeah, this microsome trail here on the top left from the fibroblast. In this video, you see one cell deposit these microsomes, another one comes and kind of gobbles them up and then kind of goes away for a little bit. The video doesn't continue, but eventually it comes back and kind of follows that trail. And on the top right there is some of Melissa Weaver's work at Vanderbilt. She has this sort of pH-sensitive diet she can use, but as you can see, she can also track matrix binding, probably microsome internalization. Here's my favorite TEM image of microsome. So the pack of all the small babies are generally around 500 nanometers or bigger. And so physically they have different properties that we can exploit to isolate them. And so you see these videos, you might think that that's just a future artifact, but recently we got one for zebrafish papers showing that these are produced in vivo and have significant effects on zebrafish development. So you can see that they're produced in zebrafish models, so we can look at these in vivo. And then over here on the right, I've just shown some testing from a lot of different cell lines, and it's relevant to your work. The one that I'm most interested in, at least in the context of this study, is this UE7 cell line, which is a glioblastoma cell line. And then just like recency, there's a paper out on retractosomes, which are just retracting fibers that are degraded. Maybe they're important, maybe they're not. It's hard to tell if I have a robust in vitro system to study them. I don't know if Jay's here, but his paper is preprint on Nature, which just came out, which I kind of supermere, exomere sauce, but also in the ECM, so before we check these out. So there's a lot of interest, I think, growing in the field, diverse types of ECM, and understanding their function. But there's really no way to study them in depth and in a highly reproducible manner. In fact, like some of those videos I showed you, they're just relying on chance. One cell deposits these matrix-bound bees, another cell comes along and internalizes them, but there's no highly controlled method of producing them. So I thought back in 2018, it was like incorporating a new approach to scalable human network patterning, ideally to study microsomes, but as you'll see in my talk, it applies to a lot of other things. And the idea was kind of to deposit EVs and then put cells on top rather than, most of the field, I think, when they do in vitro tests of the internalization, they put the cells down and then dump the EVs on top. And one thing about this was I knew I would get a lot of EVs, so I ended up using bioreactors. So the idea was, you can see those red boxes, a leading cell would be the trail, and a trailing cell would internalize those and either follow it or not. It's hard to tell when they do and when they don't, and why. And also, if you do a cancer organoid growing outwards, radially, there should be a gradient of these EVs attached to surfaces. So I thought I could make these distinct trails and also these gradients, and it would really help for the field. And so I spent a lot of time in New Zealand. I was Charisse here, but also Larry Shannon in Euphemia at University of Auckland kind of helped me work through these ideas and encouraged me to keep going. There's a lot of things you shouldn't try if you want to pattern EVs in a robust manner. Don't try micro-contact stamping. I tried reversibly bound microfluidics and like using chips to deposit them and peel them off. I think what I'm going to present is just save yourself some time. So fortunately, after like 5 years of trying different things, we were able to basically produce these patterns that are really and it's largely thanks to the global reactivity group at Ohio State. So the EVs that I'm mostly going to be showing are from these bioreactors, which I don't have a lot of time to get into, but basically we can grow really high density cells within these and get really high density EV products. And then the small EVs go from differentials, segregation sizes for the chromatography. The large EVs are going to get density gradient on following we use microsome isolation protocol. So you got microsomes on the serum, so I just follow that protocol. And for the E. coli, we'll just do tangential flow filtration and size switching chromatography. So to the application. It's pretty straightforward. So if you take a cover slip or a glass slide or something flat and semi-transparent, we coat it with PLL, which gives it a positive charge, and we put an and an SVA on top. So basically, Cornell combines the innings of your initial base layer and have an anti-fog and peg brush that's stuck to the surface. We stick a really high density photo activator on top, and then we use the DMV system, which basically rasters UV light on the surface that cleaves off a lot of that peg, exposing a positive charge. And since UV's negatively charged, we can kind of repeat this and create multi-UV patterns. So I'll show you how we do this. The great thing about the system is it's just a grayscale template that you input, so you can draw all kinds of crazy stuff, which hopefully I'll have time to show you. But first we just did a resolution test. So here I'm showing in real time how fast the exposure is. So this is highly scalable. We can pattern probably 96-volt plate in like an hour or so currently. And then those 3 EVs that I mentioned earlier, you can see we can deposit these beautiful high resolution patterns in like circles and these micro-track arrays that mimic the microzoom trains. Another cool thing is we can basically write functions. So if we use a grayscale template again, we can do Gaussian gradients, we can do linear gradients and exponential gradients. And we end up with these beautiful gradients of these different types of UVs along the surface. So ultimately I want to see how this affects cell behavior, but for now we're just quantifying how the template works relative to what we actually get on the surface based on the fluorescence intensity. It's a little bit different kind of our input versus what we get out, but we can start to correct for that in the future. Since this is an asset problem, I don't have any diagram controls because they're boring, but I do have some of these new GFP commercial EV standards. So I got the vesicular ones and the signal lines of constants here, which is some picking on here. They also have their own. But as you can see, even without using dynamic controls, we can still tell that these are actually EVs that are patterning. I do have the dynamic controls in the preprint if you really want to look at them. And then here's the gradients for those same commercial EV standards. Something we're just started working on is like, can you pattern 2 different types of EVs right next to each other? So we have been working on this for doing these arrays of different antibodies. And so we just adapted it to EVs, and so you can imagine once you put cells on here, they have to make a decision of which kind of EV they want to go to. This is very preliminary. And we're also trying to understand, like, working backwards. So we got the technique for how are the EVs actually attaching. So we took our small EVs from the big Radius 7 bioreactors, large EVs, did a characterization, and we can use time-lapse turf to actually measure the kinetics. So we were curious if the different size of EVs, because it changes their concentration, if that affects their absorption. So here you're looking at time-lapse turf of large and small EVs, and you can see that they deposit into these engineered areas of the surface. And you can see some non-specific binding, which it comes off really easily because there's PEG in those regions once we do our wash step. And we can even simulate this effect in console. So if we basically define a positively charged region of the surface, and then we put particles on top, we can simulate basically the same kinetics and then compare it to the experimental. So what we see is that we can approximate basically the binding kinetics with a really simple simulation, which we think will be helpful. And we've also done some like synthetic cationic particle controls, which I won't have time to show you. We think that this binding effect is basically a combination of size and charge. So we're showing there the zero potential values. So what applications do we have? So our lab is really interested in doing these like single EV ELISAs, but Rachel and Randy's paper kind of show that we all know tensor spins can really bias your capture, and ultimately we do diagnostics, cell typing. This can be an issue. So we thought if we have this nice non-specific capture system, what if we just label our EVs in solution, do a cleanup step, and then using our gradients, we can digitally titrate them on the surface. And so if you have a region where the EVs are way too dense for your algorithm to do the polarization, we can kind of move across the gradient until you get into a region where they're nice and single EVs. And here I'm just showing for large and small EVs, these are the Gaussian patterns, and I know this all on here, so it's pretty robust. We're not relying on any Gaussian. Back to the original inspiration, so this is actually not as developed as I would hope at this point, but we're getting there. We make these microtracks, so these are large EVs, and microzones could have the full ISO, the full validation so far. But basically, the cells on top of these trails, the video is a bit weird looking, but you can see the cells are going up and down, and we're now working through different types of controls to understand how cells behave on EVs versus just like an FBS control. And the last application, which is actually the one that's been moving along in this quickly, so we take bacterial EVs. The previous speaker kind of explained the importance of bacterial EVs. Isolate them, and we're just validating their size and morphology. If we put them in solution with neutrophils, which are kind of first responders with their immune system, and we can show that neutrophils change their morphology, they get activated. This is something people have shown before. We can quantify that. But because we have this patterning technology, we can put them on surfaces in these defined shapes. And we can, this is the first example of neutrophil swarming bacteria EVs. So we have this kind of immunoengineering platform where we can understand how immune cells interact with surface pattern EVs of all kinds. So these are controls showing no swarming and normal stuff. So the cool thing is that we're at the subcellular scale. We can start playing with geometry. So here's like a circle we pattern on a star and a square. And you can see that they swarm kind of differently depending on the edge effects. So we actually want to quantify if they're signaling to other immune cells differently based on the shape. And so on the last slide here, on the left you're going to see a circle pattern, on the right is a star. And what you see is initially they start swarming kind of evenly, and the star starts pulling in more of these neutrophils, which we don't fully understand why. There's some kind of edge effect in neutrophil signaling. We can quantify this. Which I think is, I don't really, if someone really knows the immune system, will kind of talk to me. It seems to fit in a lot of stuff. So in conclusion, LEVA, which is light induced absorption, is high resolution, tunable, rapid, and scalable. There's 2 units in there. Micropatterning of all different kinds of EVs, not specifically. There's 3 applications that illustrate future work. There's like one that I try to incorporate this in microfluidics, 3D surfaces we can pattern on, improve fractionation of microsomes and other subtypes of EVs, and how do we assess function once cells internalize these matrix-bound EVs, and then changing the chemistry to different ECM base layers. You can use like biotin and do some cool chemistry, but you just have to be really careful with EV purification. So thank you, everyone. The Center for Cancer Engineering at Ohio State funded this, along with my K99 through NFAIB. There's some EV baked goods and 3D printed EV. Going full circle, so here's this amazing figure from our paper. We can just pattern that, so it's made out of EVs. It's like your typical EV cross-section pattern out of EVs, and you get almost like a 3D effect. I think this is touch spinning here, just kind of crazy looking. And thanks for coming on this.
This was a really interesting talk. We have time for one question only.
Look, that was a great talk. I really enjoyed it. I kind of have 2 questions. Okay. With the microsome, I always find it so fascinating, and I see it in tissues as well, cells being trading, EVs behind. Now, if I were to walk from here to there and I leave globs of skin behind me, would I not fade away? How are cells able to maintain such trading signals behind them?
I don't really know. I know a lot of the energetic systems, especially in cancer, which I think is one of the more interesting applications, get completely hijacked when they're in that state. As far as cell metabolism, I'm so far on the engineering side. That's something I don't really know how to answer the question. I think it's something we could probably learn with this kind of surface patterning technique, but yeah, I don't really know. But we know that they're imbuing a whole lot of blood. You can see them in the fish, so somehow they're finding the energy to produce these things and the nutrients, but I don't fully understand it. Is it about my shirt? My shirt has EVs on it.
Thank you very much, Colin. I know a lot of people want to speak to you afterwards, and They will going to talk to you afterwards. Thank you very much. Next speaker is Jia Yi Wu, and Nanyang Technological University, Singapore, and he'll be talking about hybrid microfluidic tangential flow filtration and herringbone microstructures for rapid extracellular vesicle isolation of complex plasma.
Thank you for the introduction. Good afternoon, everyone. I'm Jia Yi, a first-year PhD student from Nanyang Technological University in Singapore. First, I'd like to thank the ISAF committee for giving me the opportunity to share my work, which will be on the hybrid microfluidic tangential flow filtration and herringbone microstructures for rapid extracellular vesicle isolation from blood plasma. So, as most of us know, EV are cell-derived vesicles that carry nucleic acids and proteins acting as key mediators of intercellular communication. The state is increased potential values of diagnostic biomarkers such as future format proteins from tumor-derived EVs, which presents an opportunity for early disease detection. So earlier my colleague has already covered some of the challenges within the clinical EV diagnostics, but I'll quickly run over them again. So during transportation from clinics to hospitals after blood drawn, hemolysis and platelet activation will occur during transportation. And during the processing of blood samples in clinical laboratories, we also have inconsistent protocols when dealing with blood samples, and then it results in incomplete or cell platelet removal. So further amplified by the high protein background, we actually have a lot of cell-derived EVs, which increase the noise for EV detection. Thereafter, different label-free methods can be used to isolate EVs, namely ultracentrifugation, size exclusion chromatography, and PFF, tangential flow filtration, which I'll be focusing on later. Many groups have also tried to develop many other methods as they see that there is an unmet need for efficient, user-friendly, and cost-effective solutions for EV solution. So in the booth, there's also Exodus, who actually have successfully done it, but they use batch processing. So zooming on TFF, comparing to other filtration, those samples perpendicular to the filter, TFF actually passes samples parallel to the filter. So it allows the retentate to remain in the feed stream as filtrate passes across the membrane. So this method is much more gentle on samples and minimizes clogging as particles can be recirculated by the filtrate feed stream. Despite its advantages, existing commercial TFF solutions still require large sample volumes, so ranging from 5 ml up to 100 ml, making them unsuitable for processing small volume plasma samples. Before I introduce my project, I would also like to briefly cover a branch of microfluidics, micromixers. So yesterday during the plenary session, Prof. Metro actually covered a very good introduction to micromixers. But in general, micromixers can be split into two types. You have active and passive micromixers. Active micromixers use external forces through additional modules to carry out highly controllable and precise mixing, whereas you have passive micromixers which use integrated geometry to carry out integrated mixing. However, they require design optimization before use. And this leads up to my work's objective, which is to develop a novel microfluidic TFF device for plasma protein depletion for EV clinical diagnostics. The device's key features are its low-cost 3D printed molds and PDMS fabricated microchannels. Thereafter, we are able to carry out small plasma volume processing with microfluidics, which is further enhanced by the integrated micromixer. Introducing the microfluidic TFF device is a 3-layered device combined using plasma membrane. So we have top and bottom microchannels sandwiching the intermediate layer, which is made up of a 50nm pore-sized membrane and a PDMS thin film. The top and bottom symmetrical serpentine channels overlay the porous membrane, enabling filtration, while the top channel features the staggered herringbone microstructure, or call it SHV, to enhance the performance. Inlet and outlet sections were split for concurrent imaging, while we have grooves to allow the accurate alignment during the bonding process. Here's the setup list of device. So syringe pumps were deployed to inject samples and buffer at differing flow rates, such that sample flow is higher. So it's around 30 to 10 microliters per minute. This will create a pressure gradient which will encourage the filtration of proteins across the membrane. The diluents from both EV and protein outlets will then be collected for downstream analysis. Imaging was carried out from the top view. So as such, samples were injected through the top channel, then images will be collected at the EV outlet. Buffer is injected through the lower channel, and proteins can be collected at the protein outlet. Following that, we captured images at 3 points: the inlet intersection, the outlet intersection before the protein outlet, as well as the EV outlet. So you can see in the right few images that we can have a very clear view of what's going on in the channels. Prior to setting up filtration, we actually passed 15 nm fluorescent beads into the device as a leakage test to ensure that it is as intended. So as you can see from the videos, the beads actually remain in the top channel, the sample channel, therefore indicating that there's no leakage within the device. Thereafter, we use 15 nm beads spiked in 0.1% DSA, Bovine serum albumin, each stimulating EVs and proteins respectively. Post-filtration analysis was carried out using NTA and BCA, and the device managed to achieve a reduced flow rate from 30 microliter per minute in the inlet, and across 30 minutes we have around 3 to 5 microliter per minute collected at the EV outlet, showing that there's a concentration. For the EV recovery, we managed to the protein removal was around 80% consistently across various time points, the recovery rate of around 7%. And we noticed similar size to concentration profiles observed at both inlet and outlet. So to enhance the filtration efficiency, we chose to use the staggered herringbone micromixer, a passive mixing microstructure, which was initially proposed by Abraham Stroud. So you can see here the herringbone microstructure. The vortices induced by staggered herringbone efficiently mixes samples under laminar flow conditions and can be easily integrated with minimal design changes or additional modules, allowing us to keep it at low cost. Prior to integration, we had to optimize this SHB micromixer for our devices' microchannels. So the parameters that we went about optimizing include the angle, depth, stagger ratio, and width and spacing. And here's a summary of the optimized parameters. So the SHB integrated channel was then compared with a featureless channel to verify the mixing efficiency. Fusing FISI BSA and PBS into respective inlets, we noticed that within a featureless channel, there's very limited mixing due to the slow diffusive mixing within laminar flow conditions. However, after integrating the SHB, we noticed that by the time the samples had reached the midpoint of the channel, we already have a fully mixed sample. Thereafter, we characterized the device with blood plasma samples, pre and post SHB integration. Through the processing of 20 times diluted blood plasma, we obtained similar protein removal rates of around 70% with lower protein concentration with the SHBs. CD9 positive recovery rates was higher at around 35% compared to 15%, with higher EV outlet concentration with the SHB. Overall, the SHB integrated device demonstrated greater EV purity as compared to the one without the SHB. In summary, we have developed a hybrid microfluidic tangential flow filtration and herringbone microstructures for rapid extracellular vesicle isolation from blood plasma. which is low cost, capable of processing small plasma volumes, as well as enhanced filtration through the passive microwave cell. Looking forward, we plan to integrate this device with the device introduced by my colleague Sean earlier to develop an automatic blood processing device for POC, point-of-care EV diagnostics. And I'd like to thank my supervisor, as well as my colleagues, collaborators, and funding sources. And these are our groups.